There is a lizard on a small Indonesian island right now that weighs as much as a grown man, moves with a stillness so complete it is almost indistinguishable from the fallen logs around it, and carries in its mouth a biological arsenal so sophisticated that scientists spent decades arguing about what it actually was. The Komodo dragon is the largest lizard alive on Earth, and its deadliness is not simple. It is layered, refined across millions of years into something that works so well and so quietly that its prey often does not fully understand what has happened until it is too late. For a century, the story told about the Komodo dragon was the bacteria story, that the saliva of the dragon harbored such a toxic consortium of bacteria that a single bite guaranteed infection and eventual death in any prey that escaped. It was a dramatic story. It was also, as it turned out, largely wrong. The truth behind the Komodo dragon's lethality is stranger and more precise than any bacterial theory could account for, and it involves venom glands, anticoagulant proteins, and a hunting strategy of such patient, architectural efficiency that it has no real parallel among any other reptile alive today. Tonight, we will follow the Komodo dragon gently through all of it, through the real story of its evolution on islands that shaped its biology in ways no mainland lizard has experienced, through the anatomy of a killing system that operates across hours and kilometers, and into the quieter questions about a mind that is more capable than its cold-blooded reputation has ever given it credit for. So, let the room settle around you into a still, warm quiet, the kind of quiet the dragon knows intimately, the quality of stillness that is not absence but attention, and drift with me to the island of Komodo, where the largest lizard on Earth is already watching something it will not need to hurry to catch. To understand why the Komodo dragon became so large and so deadly, you have to begin not with the dragon itself, but with the islands it lives on. Because the Komodo dragon is fundamentally an island animal, and island biology follows rules that are different from those that govern mainland evolution in ways that produce outcomes no one would predict from general principles alone. The Komodo dragon is found wild only on 4 islands in the Indonesian archipelago: Komodo, Rinsa, Gili Motong, and the western tip of Flores. A geographic range so small that its entire wild habitat would fit inside a modest metropolitan area. On these islands, the dragon evolved in an ecological context that had no equivalent anywhere on the mainland, and that context is what produced the animal it became. The most important ecological fact about the Lesser Sunda Islands, where Komodo dragons live, is that for most of their evolutionary history, the dragons were the apex predator. There were no large mammalian carnivores competing with them for prey. No tigers, no leopards, no wild dogs capable of displacing them from a kill or suppressing their hunting activity. The prey community included deer, wild pigs, water buffalo, and goats, large mammals that would represent significant prey items for any predator. But the dragons had no competition for these resources beyond each other. In this ecological vacuum, the selection pressure was for size. A larger Komodo dragon could kill larger prey, could defend kills against rival dragons, and could survive the periods between successful hunts on stored fat reserves that only a large body can accumulate in meaningful quantities. The island acted as an evolutionary incubator, selecting for greater and greater size over millions of years, until the animal reached the dimensions we observe today. The evolutionary origin of the Komodo dragon has been illuminated by fossil evidence, that places the origin of the Varanus lineage, the monitor lizards to which Komodo dragons belong, in Australia, rather than in Indonesia, where they now live. Fossil monitor lizards of impressive size, including the extinct Varanus priscus, sometimes called Megalania, which may have reached 6 meters in length and represented the largest terrestrial lizard in Earth's history, roamed Australia during the Pleistocene. Genetic and fossil analysis suggests that the ancestors of the modern Komodo dragon dispersed from Australia northward into the Indonesian archipelago, where they eventually became isolated on the Lesser Sunda Islands and continued evolving in the specific conditions of those islands. The Komodo dragon is, in this sense, a remnant of a much grander radiation of giant monitor lizards that once occupied a broad range, subsequently going extinct across most of that range, while persisting on its island refugia to the present day. The prey community that the Komodo dragon evolved alongside on these islands has shaped its hunting biology in ways that are worth appreciating in detail. The Javan rhinoceros and the extinct pygmy elephant stegodon once shared these islands with the dragon, and stegodon in particular was an important prey species for large Komodo dragons during the Pleistocene, its size requiring the kind of coordinated, multi-individual hunting and the specific anatomical killing adaptations that we now see in the dragon's biology. As stegodon and the pygmy rhinoceros went extinct, the dragon shifted its primary prey to the deer and buffalo that remain. But the killing apparatus refined against those ancient large prey remains fully functional against the current prey community and is arguably more than sufficient for the task. The Komodo dragon is, in biological terms, slightly overdeveloped for its current prey, carrying a set of weapons sharpened against animals considerably larger than anything it regularly hunts today. The island ecology of Komodo has also produced a social structure and feeding behavior in the dragon that is unlike that of most other large reptiles. Komodo dragons are not solitary in any simple sense. They gather at carcasses in organized feeding hierarchies, in which body size determines access, with the largest males eating first and smaller individuals, females, and juveniles waiting at the periphery or feeding on the scraps after the dominant animals have finished. These feeding aggregations involve a complex of postural signals, threat displays, and occasional physical confrontation that suggests a social awareness and individual recognition capacity that goes beyond what most people expect from a reptile. The same island conditions that drove the evolution of large body size also drove the evolution of the social intelligence needed to navigate feeding competition in a population of large predators sharing a limited geographic space. The story of Komodo dragon venom is a story about how scientific consensus can persist for decades on the basis of a compelling narrative rather than rigorous evidence, and about how the actual biology, when finally examined carefully, turns out to be more interesting than the story it replaced. From the early 20th century, when Komodo dragons first came to the attention of Western science, the explanation for why prey bitten by a dragon and initially escaping died reliably within days was bacteria. The dragon's mouth, it was said, harbored an extraordinary diversity of pathogenic bacteria, maintained by the rotting meat stuck between the teeth and the warm, moist environment of the oral cavity. A single bite delivered enough bacterial contamination to guarantee septicemic infection in the prey, which would weaken and die over days while the dragon tracked it patiently using its extraordinary sense of smell, eventually finding the dead or dying animal and consuming it. This bacterial theory was accepted so widely and repeated so consistently that it became embedded in popular accounts of Komodo dragon biology as established fact. The problem was that it had never been subjected to the kind of rigorous experimental testing that genuine scientific consensus requires. When Brian Fry and his colleagues at the University of Melbourne examined the bacterial theory systematically in the late 2000s, they found that the bacterial populations in Komodo dragon mouths, while diverse, were not substantially more pathogenic than those found in the mouths of other large carnivorous reptiles and mammals that eat whole prey and do not subsequently kill their bitten prey through infection. Komodo dragons kept in captivity and fed sterile prey maintained their hunting effectiveness without exposure to the rotting meat environment that the bacterial theory required. The bacterial explanation was not supported by the evidence when that evidence was actually collected and examined. What Fry and his team found instead, through anatomical dissection and chemical analysis, was a pair of venom glands in the lower jaw of the Komodo dragon, producing a complex mixture of proteins with anticoagulant, antiplatelet, and blood pressure-lowering properties that, when delivered into a wound through the dragon's serrated teeth, would cause profound and rapidly progressing hemorrhagic shock in prey that managed to escape the initial attack. The venom does not kill quickly by itself. It works through a suite of biochemical effects that together prevent the prey's blood from clotting normally, dilate blood vessels, lower blood pressure, and induce a state of physiological shock that weakens the animal progressively over hours and makes it increasingly unable to sustain the flight from a predator that its initial escape momentum might otherwise have allowed. The dragon then follows, tracking with its forked tongue and the vomeronasal system, and finds the prey in a state too compromised to escape a second attack. The venom of the Komodo dragon belongs to a lineage of toxin production that the research team traced across the monitor lizard family and further into the broader squamate reptile lineage, including snakes, suggesting that venom production evolved once in a common ancestor of monitors and snakes and has been retained or lost independently in different lineages. This finding, which was controversial when first published, has been substantially supported by subsequent research and represents a significant revision of the understanding of venom evolution, suggesting that the venom system is much more ancient and more broadly distributed across reptiles than previously recognized. The Komodo dragon, in this framework, is not a uniquely venomous aberration among monitor lizards, but a member of a venomous lineage whose toxin system has been expressed to different degrees in different species. The teeth of the Komodo dragon work in concert with the venom delivery system in ways that maximize the effectiveness of a bite. The teeth are large, serrated on both edges, and curved backward, an architecture that functions like the serrated edge of a steak knife, tearing through tissue as the jaw closes and creating a wound channel substantially larger than the tooth cross-section alone would produce. The backward curvature of the teeth makes it mechanically difficult for prey to pull a bitten limb out of the mouth without enlarging the wound further, and the serrations on the tooth edges maximize tissue damage per unit of jaw force applied. The gums surrounding the teeth are arranged to retract and bleed readily during feeding. A design that was initially cited as evidence that the bacteria-laden blood in the mouth contributed to infection, but which Fry's research suggests may actually facilitate venom delivery by allowing the venom seeping from the lower jaw glands to mix with the blood flowing from the gum tissue and be carried into the wound via the serration channels in the teeth. The mouth is a coordinated weapon system with teeth, gums, and venom glands all contributing to a delivery mechanism more sophisticated than any of them could achieve in isolation. The Komodo dragon's forked tongue is not merely a sensory organ. It is the primary interface between the animal's nervous system and the chemical landscape of its environment. And its capabilities are so far beyond what the word smell conventionally implies that the comparison to human olfaction does not really capture what the system does. The tongue collects airborne and surface-borne chemical molecules from the environment with each flicker, and the two tips of the fork collect samples simultaneously from two slightly separated points in space. These samples are delivered to the vomeronasal organ, also called Jacobson's organ, a chemosensory structure in the roof of the mouth that processes the molecular information and sends it to dedicated neural circuits in the brain. By comparing the chemical concentrations at the two fork tips, the Komodo dragon's brain can determine the direction of a chemical gradient, following it toward the source, the way a person might follow a smell toward its origin, but with a precision and range that far exceeds anything the human nose can achieve. The practical hunting application of this chemosensory system is the ability to track prey or locate carcasses across distances measured in kilometers. Komodo dragons have been documented following scent trails of blood and decomposition across distances of up to 10 kilometers, navigating through the dense, dry scrub of Komodo Island with a directional precision that would be remarkable in a mammalian predator with a comparably sophisticated olfactory system. The tongue flickering that Komodo dragons perform continuously while moving is not a nervous habit or a simple atmospheric sampling. It is active, directed environmental scanning, with the dragon adjusting the direction and frequency of its tongue flicks in response to the chemical information being collected, steering toward chemical gradients of interest and away from gradients that indicate danger or unproductive areas. The dragon builds a chemical map of its environment through this continuous sampling and navigates it with the same purposefulness that a visually oriented predator navigates by sight. The integration of chemical information with visual information in the Komodo dragon's hunting behavior reveals a sensory sophistication that is often underappreciated in animals that share the popular reputation of reptiles as simple, reflexive creatures. Komodo dragons have been observed using visual information to identify and ambush prey at sites where they have learned, through experience, that prey animals regularly pass, waiting motionless in ambush positions for hours or days before a suitable target presents itself. This combination of experienced spatial knowledge, the memory of where prey travels, with the chemical tracking that allows the dragon to locate prey it cannot see, produces a hunting system that operates across multiple timescales simultaneously, from the seconds of the actual strike to the hours of the ambush wait to the days of the post-bite tracking. The visual system of the Komodo dragon is itself considerably more capable than the reptilian reputation for poor vision would suggest. Komodo dragons have good color vision and can see clearly at distances of up to 300 meters, which in the open savanna and dry scrub habitat of their islands provides substantial visual hunting range. Their eyes are positioned to provide a degree of binocular overlap in the forward visual field, supporting the depth perception useful for judging strike distances accurately. In low light conditions, the visual system becomes less useful, and the chemosensory tongue system takes over as the primary information gathering mechanism. The transition between visual and chemical primary sensing as light changes through the day is a behavioral flexibility that reflects the integration of two independent sensory systems rather than the reliance on any single one. The hearing of the Komodo dragon has historically been described as poor based on early studies that found limited responsiveness to airborne sound. More recent research has revealed a more nuanced picture. While Komodo dragons are indeed less sensitive to airborne sound than mammals, they are sensitive to low-frequency vibrations conducted through the ground and through the substrate they are in contact with. And these substrate vibrations may provide information about the approach of large animals that the airborne sound system would miss. The full sensory picture of the Komodo dragon is therefore one of a multi-channel information gathering system with chemical, visual, and vibrational inputs integrated into a behavioral output that is considerably more sophisticated than the simple reactive predator of popular imagination. The hunting strategy of the Komodo dragon is built on patience in a way that reflects both the biology of the venom and track system and the specific ecological context of an island predator that has no competition for its prey and no urgency driving a rapid resolution to each hunt. A Komodo dragon in hunting mode is not necessarily moving. It may be motionless for hours in a chosen ambush position, its cryptic coloration of gray-brown scales providing camouflage against the dry, rocky terrain of its island habitat. Its metabolic rate, low in the way of all reptiles, but specifically calibrated to the ambush predator lifestyle, allows it to maintain alertness without the energetic cost that sustained muscular activity would impose on a mammalian predator of equivalent size. It is not waiting in the way a tired animal rests. It is waiting in the way a trap is set. The actual strike of the Komodo dragon is explosive and brief, deploying muscular power that allows the animal to reach a running speed of approximately 20 kilometers per hour over short distances, sufficient to close the gap to an inattentive deer or goat from ambush range before the prey's escape response can fully engage. The bite itself takes a fraction of a second, and the dragon's goal in the strike is not to kill immediately, but to deliver the venom-loaded wound in a location likely to cause maximum hemorrhagic effect, typically to the leg, throat, or underbelly of the prey, where major blood vessels are accessible through relatively thin tissue. Having delivered the bite, the dragon will typically release the prey rather than attempting to maintain grip and wrestle it into submission, a strategy that makes sense given that a large prey animal fighting to escape could cause significant injury to a dragon that tries to hold on. The release is not failure. It is the first step of a two-stage hunting process in which the initial bite is the investment and the tracking is the collection. After the initial bite, the dragon returns to its routine activity of tongue-flicking environmental scanning, but now with the specific chemical signature of the wounded prey's blood trail added to the environmental chemical landscape it is reading. The prey, initially fleeing on the adrenaline of escape, will travel some distance before the combined effects of blood loss, venom-induced anticoagulation, and progressive hypotension begin to compromise its physiological function. As its blood pressure falls and its ability to sustain muscular activity diminishes, it slows. The dragon, following the chemical trail of blood and physiological distress with its tongue, closes the distance gradually. In cases where the prey is a large animal like a water buffalo, this tracking phase may extend over hours and several kilometers, with the dragon following the weakening animal until it is no longer capable of flight. At that point, the dragon feeds, and if the carcass is large enough, it may return to feed over multiple days, consuming an enormous proportion of its own body weight in a single extended feeding event. The thermoregulatory biology of the Komodo dragon provides the metabolic foundation for this patient hunting strategy. As an ectotherm, the dragon does not generate body heat metabolically in the way that mammals do, and its activity level, hunting performance, and digestive efficiency all depend on its body temperature, which it regulates by moving between sun-exposed and shaded areas throughout the day. In the cool, early morning, the dragon basks to raise its body temperature toward the optimal range for activity, typically between 35 and 40 degrees Celsius. As the day heats up and the ambient temperature rises, the dragon seeks shade to avoid overheating. The active hunting period is therefore concentrated in the mid-morning and early afternoon hours, when body temperature is in the optimal range, with the metabolic cost of that activity being a fraction of what an equivalent-sized mammalian predator would expend for the same behavioral output. A Komodo dragon can survive on approximately 1/10 the food that a mammalian carnivore of equivalent size requires, because the baseline metabolic cost of maintaining body temperature is absent, and the resting metabolic rate is correspondingly low. This metabolic economy is what makes the days-long wait between hunting opportunities viable, and what allows the dragon to devote a large fraction of its energy intake to growth, rather than to the maintenance metabolism that consumes the majority of a mammal's caloric intake. The Komodo dragon's ability to reproduce parthenogenetically, producing offspring from unfertilized eggs without the involvement of a male, is one of the more biologically remarkable features of a species that is already remarkable in many other ways. Parthenogenesis has been documented in captive female Komodo dragons that have been isolated from males for extended periods, producing viable offspring that are genetically derived entirely from the mother. The offspring produced by parthenogenesis are not genetically identical to the mother, because the mechanism involves the duplication of half the mother's genetic material rather than simple cloning. But they represent a viable reproductive option when no males are available. Researchers have proposed that this capability may be particularly relevant for the colonization of new islands, where a single female washed ashore from a driftwood raft could potentially establish a new population through parthenogenesis, producing male offspring initially, and then mating with those males in subsequent generations to restore sexual reproduction. Whether this scenario has actually played a role in the historical distribution of Komodo dragons is speculative, but the capability for it exists and has been demonstrated in captivity. The cognitive capabilities of Komodo dragons have been studied with increasing rigor as researchers have moved beyond the assumption that reptilian behavior is primarily reflexive and largely uniform within a species. The picture that has emerged from field and captive studies is of an animal with genuine individual behavioral variation, the capacity for learned behavior that transfers across contexts, and a social awareness sophisticated enough to support the kind of feeding hierarchy and individual recognition that characterizes Komodo dragon aggregations at carcasses. None of these findings require attributing human-like cognition to the dragon. They do require moving beyond the stereotype of the reptile as a behaviorally simple stimulus-response machine that makes decisions through nothing more complex than fixed action patterns and simple conditioning. Individual Komodo dragons in captivity show personality differences that are stable across time and contexts, with some individuals consistently bold in approaching novel objects and unfamiliar situations, while others are consistently reactive and avoidant. These consistent individual differences, meeting the standard criteria for animal personality, have been documented across sufficient numbers of individuals and sufficient time periods to be treated as a genuine population-level phenomenon rather than individual curiosity. The existence of personality in Komodo dragons is not surprising from a theoretical standpoint, since the ecological context of an island apex predator facing varied hunting challenges and social competitive situations would be expected to generate selection for behavioral flexibility and individual strategic variation. But it adds a dimension to the animal's behavioral biology that the simple reptile stereotype has no room for. Komodo dragons in captivity show spatial learning and memory capacities that allow them to navigate complex enclosures, locate specific food sources reliably across multiple trials, and update their spatial knowledge rapidly when resource locations change. These capabilities are consistent with the field behavior of wild dragons that learn and remember the locations of productive ambush sites, water sources, and carcass locations across their territories, returning to these sites preferentially and apparently adjusting their spatial decisions in response to the outcomes of previous visits. The accumulation of this kind of spatially organized experiential knowledge over the lifespan of an animal that can live for more than 30 years in the wild represents a behavioral repository of ecological information that would give an experienced dragon a meaningful competitive advantage over younger, less experienced individuals in the same territory. The play behavior documented in juvenile Komodo dragons in captive settings is perhaps the most unexpected finding in the study of Komodo dragon cognition, because play has historically been associated with mammals and birds rather than reptiles, and its presence in a reptilian species challenges the taxonomic boundaries that the concept had previously respected. Juvenile Komodo dragons at the National Zoo in Washington were documented repeatedly engaging in object play, interacting with novel items provided by keepers in ways that were not associated with feeding, escape, or any other obvious functional goal. The behavior was voluntary, repeated, and specifically targeted at novel objects rather than familiar ones, meeting the standard criteria for exploratory play. These observations do not make the Komodo dragon cognitively equivalent to a mammal that plays. They do suggest that the behavioral flexibility and curiosity that motivate play are not exclusive to warm-blooded vertebrates, and that the cognitive capacity to engage with the environment for its own sake may be more broadly distributed across the vertebrate lineage than the traditional view has assumed. The conservation status of the Komodo dragon reflects both the animal's remarkable resilience as a species, adapted to conditions that would challenge most large predators, and the specific vulnerability of a species whose entire wild existence is confined to 4 small islands. The total wild population is estimated at between 3,000 and 5,000 individuals, a number that might seem substantial until you consider that this entire population is distributed across an area smaller than many single wildlife preserves elsewhere in the world, and that the islands are subject to the threats of tourism pressure, habitat degradation, sea level rise, and the climate-related changes that are affecting the frequency and distribution of dry season fires across the archipelago. The Komodo dragon was reclassified from vulnerable to endangered on the IUCN Red List in 2021, with the primary driver of the reclassification being climate change projections that suggest a significant reduction in suitable habitat on the dragon's islands over the coming decades, as rising sea levels inundate the low-lying coastal areas where the dragon's prey concentrates and the dragons hunt most effectively. An animal that survived the extinction of the Pleistocene megafauna, that outlasted the giant lizards of Australia, that persisted through the climate fluctuations of the ice ages, now faces a threat whose timescale is measured in decades rather than millennia, and whose source is distant from the islands where the dragon lives. The island is very quiet at this hour. The dragon that has been still since before the light changed is still, in its chosen position among the rocks, tongue flickering occasionally into the cooling air, reading the chemistry of the night with the patient attention of an animal that measures time in ways that have nothing to do with urgency. It will hunt again tomorrow, or perhaps the day after. The island will provide, as it has always provided, in its own slow way, and the dragon will wait in the particular quality of stillness that is not sleep and not rest, but something closer to readiness, a long, warm, reptilian attention to the world that requires nothing from the world in return except that it continue to offer, eventually, what the dragon is prepared to take. You have been tonight inside a killing system built not from fury, but from patience, inside a venom that works through chemistry rather than violence, inside a tongue that reads the world in molecules rather than light. The Komodo dragon is the product of an island, of a prey community that no longer exists, of millions of years of selection for the particular efficiency that only a large, cold-blooded, unhurried apex predator can achieve. It is, in every feature of its biology, exactly what it needed to become, and it rests in that completeness the way all things rest when they are exactly where evolution put them. Let the warmth of this room settle around you now, with the slowness of an ectotherm absorbing heat from a warm surface. The dragon is still on its island. The night is cooling around it, and the extraordinary, ancient, patient biology of the most formidable lizard alive is resting in the dark, waiting with complete confidence for whatever the morning brings.